This tutorial has been updated to reflect modern methods for HIV detection, beginning with a demonstration of the ELISA capabilities of KCIT version 705.
The first thing we need to do is to click on the protein button on the silver button bar, which is located on the left. And then here is the open dialog box. Locate the cases folder, then double click on it. Locate the infectious disease folder. And we will use HIV as an example. And then the example I would like to use is located in the US folder. So I'll double click on that. And ANA is the case that I'm interested in right now for demonstration purposes. Inside the ANA folder are 3 folders, and the one of interest right now is the one that includes sequences for the ELISA and Western. And since I had originally clicked on the protein button, that's what I want to select. I could either click that and click open, or I could double click, but in this case I'll just click open. And you'll notice that that protein file is now open. It appears in the open in process window. It contains characters in 6 separate sequences. And we can see those sequences if you clicked the sequence button. You don't actually have to do this to run the ELISA, but just for demonstration purposes, here are the HIV protein sequences that we will be working with. The next thing we need to do to run an ELISA is to open up the patient samples that may or may not contain antibodies to HIV. So I will click on the antibody button on the silver button bar. And then whatever one happens to be at the top, in this case it's trimester 1. I will click on that to highlight it. I'll move down to the one just above the protein in this case. You just want to select all those antibody sequences and click open. And they will appear in the open in process window. So now what you should do is click on the protein to make sure that that is activated. And that's what will align the wells of the ELISA.
As an aside, if this was a 4th generation ELISA, the wells would also be coated with antibodies to the P24 HIV capsid protein, making it considerably more accurate than earlier generations of ELISA.
Then you select the first antibody sample, whatever it happens to be. In this case, it's on a trimester 1. Hold down the Shift key and select the last antibody sample. Highlight all of those, and then use the Quick Load and Run button and go down to ELISA. And option 1, load the same protein and different antibodies. There are other cases that where option 2 would be appropriate, but in this case option 1 is appropriate. So that's what I will select. This is a quick load procedure, so it automatically loads the ELISA plate, but there's a way of doing this stepwise as well that I'm not going to demonstrate in this tutorial. So now that it's loaded, all we have to do is click the Run button here to run the ELISA. And you'll notice that the wells were automatically labeled when you use this procedure. And to make this a little more realistic, you normally would read an ELISA plate on a light box. Go to the Options button, click that, select ELISA, and what we will use in this particular case are absorbance values, optical density. So I will select that, and you'll notice that now these optical density values have appeared. I'm not going to try to interpret this right now, but I would say that for something to be considered positive, it should be at least twice the negative control. And here you can see the one that's not very clear-cut is the boyfriend. It's fairly faint, and it's just about twice the negative control. So we're a little unsure of that. And remember, the ELISA is just the first test you would do if somebody came in to be tested for the presence of HIV. What we really need to do, if we're unsure of our results here, is to run a more definitive test, which is the Western blot. And now I can very quickly run a Western blot. Again, I will demonstrate the auto load features. Go ahead and re-click on protein. You always want to redo this clicking before each procedure. So click on the protein to activate it. Do a shift click to include all of those antibody samples. and then go to quick load and run, and down to Western blot. And just like for the antibody, we want to load the same protein and different antibodies. So I will select that. It will automatically load the wells. And now I could go to the click load and run button down to Western blot, and I can go ahead and run it after loading the samples. Now I could have just selected this option to do it all in one step, but I didn't. I selected this option and this option. So now we have to actually run the Western blot, so I will select that. Unlike the ELISA, the blot is not automatically labeled. So I can quickly do that by highlighting 1 antibody trimester 1, copy into the gel label, 2 antibody trimester 2, copy, 3 antibody baby, 4 antibody boyfriend, 5 the negative control, click to the left of the 2 and space that, click to the left of the 3 and space that, click to the left of the 4, hit the enter key, click to the left of the 5 and then space that over, and then do the same thing for 6.
The ELISA plate has been lined up with the Western blot so we can compare the results. Keep in mind this is a historical example from a number of years ago when Western blots were considered the gold standard for HIV detection. That's no longer the case. There are other procedures used, one of which will be demonstrated later. Just use your imagination, put yourself back in time, and this is the way things would have been interpreted a number of years ago, especially towards the beginning of the AIDS epidemic. And it does show the value of Western blots, which is still a very commonly used research technique for other purposes. Anna in trimester 1 in this example had a relatively low OD value, indicating that she was negative for HIV during trimester 1. And you can verify that looking at the Western blot. There's no antigen antibody reactions here. No bands are showing in lane 1. In trimester 2, though, she had a higher OD value with a strong positive result for the ELISA. All 6 bands were showing for the Western. And the criteria back then was if bands 2, 4, and 6 were showing, that was considered definitive. So back then she would have definitely been considered definitive for HIV. Her baby had a strongly positive ELISA, but notice only 2 of the 6 bands are showing in lane 3 of the Western blot, and it doesn't match the CDC's criteria back then. So the baby would have been considered indeterminate and additional testing would have been done. For lane 4 for the Western, all 6 bands are showing, and that's interesting because that's the boyfriend's result. And if you look at the ELISA, notice an OD of 0.222, compare that to the negative control of 1.02. So that's just a little bit over twice the negative control, so that's right on the borderline for the ELISA. But for the Western blot, which was considered definitive, that indicates that he definitely had an HIV infection. And then for lane 5 of the Western is negative, which you would expect because that's the negative control. Lane 6 is positive, which you would also expect because that's the positive control. And now we're going to come back to the present and look at one of the techniques that's used for a more precise HIV diagnosis. That technique is abbreviated RT-qPCR. And in this technique, viral RNA from a patient's blood sample is reverse transcribed into cDNA and is put into a specialized thermocycler so that the resulting cDNA can be quantified. So we'll use the DNA button to open samples. Go to the cases folder, the infectious disease cases, HIV, US, ANA, and now we'll go to the viral load folder. And we will be opening 3 DNA files. These would actually be cDNA that had been reverse transcribed from viral RNA. So these are the 3 samples. So that first step is not shown. We're just starting with the DNA itself for this example. Since it's PCR, we need to open a primer and then simply shift-click the 3 ANA samples from 1 month, 3 months, and 6 months, because we're trying to follow her viral load over time, assuming she's under treatment. Go to the Quick Load Run button, down to 96-well PCR. This is really 96-well qPCR. Load the primer and the DNA samples, and then click the Run button. Now, you really wouldn't be seeing this result inside the machine, but this would indicate a certain point in time on the thermocycler, showing that in 1 month's time, Anna had a fairly substantial viral load apparently from this positive result, and then it seems to fade over time. To see what the actual numbers are, go to the Options button, down to 96-well PCR and viral load. The results indicate that her therapy is quite effective because the numbers go from 55,000 units to 5,000 down to 150. We're going to demonstrate a more realistic depiction of the qPCR process using that feature of KCIT. The data that we will be using is accessed via the qPCR button on the silver button bar. Open file containing qPCR fluorescence chart data. And the data is called qPCR Honeybee. That's because this data came from an actual research project at the University of Wisconsin, River Falls. It's very difficult to get a hold of fluorescence data for HIV patients, so we will use this as a surrogate data set. And we'll only be using part of the data set. So I click the Open button to open the data. We don't need these windows open. And I'm going to move the chart up so that it's visible on your screen. Next, click the Load button right here. And that will load all 96 of the wells. Go back to the qPCR button and select Run qPCR for orange wells only. So this is what the qPCR software would generate for an actual machine. You end up with these curves that start out growing very slowly, then grow exponentially, and then level off, like a logistic growth curve. That's because with each step of the thermocycler, the DNA is being amplified. It's a series of heating and coolings. So not until 20 or 22 do you get enough DNA where it starts growing exponentially, and then it will level off. Where each one of these curves intersects this green line, which is called the threshold, that number here is the CT value. And if I'm going to just move my cursor over some of these. But so well C6, for example, has a CT value of 24.47. In other words, about 24 and a half cycles until there was enough cDNA in that sample to hit that threshold. So that's well C6, 24.47. If we look at well C6 right here, 24.47. So that's where those numbers come from. It's called the CT value. And the lower the CT value, the more cDNA there was in that sample. That means that it will start that exponential growth phase earlier because there was more cDNA to start with. The bigger the CT value, the less cDNA there was in the original sample. We'll now use the Honeybee dataset as a surrogate dataset for HIV samples. Assume for the sake of argument that well C6 is one of Anna's samples from the HIV case. And let's further assume that was one of the samples that was done at 3 months. see what would happen if we just ran qPCR for that one sample, go to the button, run qPCR for purple wells only. And it gives a value of 24.47 as we showed before. But now you can see this single curve, and you can see where it crosses the threshold. If you hold your cursor, it says 24.53. That's because it depends on which pixel that cursor hits. But the actual value is 24.47, so this value will be close to that value. Normally these are run in triplicate, so we will select 2 more wells. Back to the qPCR button and run that for purple wells only. And now you can see that they are pretty tightly clustered together, so we will use that average value a little bit later of those 3 CT values. So again, that was Anna's samples at 3 months. Let's do 3 samples at 1 month, and for that I'm going to select E4, E5, and E6, as you can see over here. Let's go ahead and run that for purple wells only. These are more to the left, indicating that there must have been more viral DNA in our samples at that point in time because it crossed the threshold earlier than the sample at 3 months. So these again would be the 3 triplicates from 1 month with a value somewhere around 22.6 or so. These are the 3 actual values. We'll average those and use those average values later when we look at a standard curve. And finally, we need to do her samples at 6 months. So we'll select B7, B8, and B9. Go to the qPCR button and say purple wells only. And now we have all 3 sets of data. Notice these are not as tightly clustered together, but all 3 of them are more to the right of this one and this one, indicating that there was less viral RNA in her sample at 6 months. To determine viral load from this data, we need to generate a standard curve. First thing I will do is go to the clear menu and say clear everything. Bring this one up. Select the qPCR button, open file, and this time select the standard curve dataset. Click the load button, run qPCR for orange wells only. This standard curve was run with duplicates, so you'll notice that they are consistent with one exception, and it's this one right here. So these are very close, as are these. This looks like an outlier. And notice it is well D2. Looking at the amplification plot here, this is the line for well D1 and D2 is way over here. So that's clearly an outlier. Something was wrong with the experimental procedure. If you look at the standard curve, which is going to plot the initial quantity copies versus the CT, you can see that this is clearly an outlier. To be very careful about outliers because sometimes they can be the most important data point in a set, but in this case it's clearly something amiss with the experimental procedure. So we're going to rerun that for all of the wells except for well D2, which is the outlier. To do that, first I need to go back to the amplification plot, and before I got to the standard curve by clicking this button. But we need to be on the amplification plot, and I'm going to go to well D2 and select it to turn it purple, and then run qPCR for orange wells only. And you'll notice the outlier is no longer there. Going back to the standard curve, you can see that that data point is missing. And now what we want to do is plug in the average CT values that we generated before, the average of those 3 values for 1 month, 3 months, and 6 months into this box. To do this, we're going to need to magnify the chart, because if you leave it at this size, some of these numbers are going to be cut off. So I'm going to click this magnifying button, bring this up. So the number I'm going to put in here for Anna at 1 month, using again the honeybee data as a surrogate for Anna's data, the average value of the CT for E4, E5, and E6 for those 3 wells was 22.64. So I'm going to type that in. And you can see the result, the number of copies, 1.745E plus 0.3. That's scientific notation, and it means that the decimal point should be moved 3 places to the right. So that would be 1,745 copies. 3 months, the average of the CT values for cells C4, C5, and C6, I calculated that as 24.43. Giving interpolated copies of 5.149E plus 0.2. So move the decimal point 2 units to the right. That'd be 514, or if you round up, 515. And then finally, for Anna at 6 months, if you recall, that was cells B7, B8, and B9. The average for that I calculated as 25.68. Moving that 2 units to the right, that's 219. So at 1 month, there were 1,745 copies. At 3 months, 515. And at 6 months, 219. So the viral load is decreasing over time. Even though those are unrealistically low values for a real HIV infection, this example does show how qPCR can be used to determine viral load.